Hello, welcome to Medbits, your exam savior. Today, let's discuss another important topic, that is counter current mechanism from juxtaglomerular apparatus. This is third part of JGA from Physiology. Those who haven't seen the part 1 and 2, please do watch it also. In part 1, we discussed about the definition of JGA, the parts of JGA and functions, and part 2, we dealt with short-term and long-term blood pressure regulation, and this is on counter-current mechanism. If you ask me, all these three parts are equally important in the exam point of view. In counter-current mechanism of JGA, we are going to talk about the case of a single nephron, its glomerular filtration and juxtaglomerular apparatus, and its connected distant convoluted tubules and the capillaries. Let's look into the definition of countercurrent mechanism. The method used by the kidneys to concentrate urine is called the countercurrent mechanism. It's very simple, right? The countercurrent mechanism takes place in juxtamedullary nephron. There are various steps involved in concentrating urine, or let's say for countercurrent mechanism. Before that, let me just tell you, please have a look into the diagram and learn to draw it. And this is known as a loop of Henle, which is coming from a nephron. And this is the capillary which is surrounding loop of Henle's, and it is known as vasa recta. This is about a single nephron, and we have such millions of nephrons in our kidney system. Step 1 is assume that Henle's loop is filled with 300 milliosmolar per liter concentration equal to that which leaves the proximal tubule. See, here is 300 milliosmolar per liter concentration. And the second step is the thick ascending limb active ion pump on Henle's loop reduces the concentration inside the tubule and increases the interstitial concentration. This is the descending limb of Henle's loop, and this is the ascending limb of Henle's loop. You can see the thickness difference here. This is the thick one, and here is happening the step number 2, the thick ascending limb. What happens there? It active ion pump on Henle's loop is reducing the concentration inside the tubule, and then what happens? And then increases the interstitial concentration. Interstitial means that is the cells between the loop of Henle's and vasa recta. And let's look into step 3. Due to osmosis of water out of the descending limb, see, you can see here H2O, the tubular fluid in the lower limb and the interstitial fluid rapidly achieve osmotic equilibrium. The additional fluid flow from the proximal tubule into the Henle's loop, which allows the hyperosmotic fluid produced previously in the descending limb to flow into the ascending limb. And the step number 5 is additional ions were pumped into the interstitium with water remaining in the tubular fluid until the osmotic gradient is reaching to 200 milliosmolar per liter. And step number 6 is again the fluid in the descending limb comes into equilibrium with hyperosmotic interstitial medullary fluid. Interstitial medullary fluid is this area between loop of Henle and vasa recta. And as the hyperosmotic tubular fluid from the descending limb flows into the ascending limb, the more solute is continuously drained out of the tubule and deposited in the medullary interstitium. Now we have come to the last step, that is step number 7. These steps are repeated over and over until step number 6 are repeated over and over, with the net effect of introducing more and more water soluble to the medulla. With ample time, this cycle slowly traps the solutes in the medulla and multiplies the concentration gradient formed by the active pumping of ions from the thick ascending limb, eventually increasing the osmolarity of interstitial fluid to 1200 to 1400 milliosmolar per liter. Let's look into the details of the countercurrent mechanism in the Henle's loop once again. The nephrons involved in the concentrated urine formation stretch all the way from the kidney cortex to the medulla and are followed by vasa recta. The movement of filtrate is in opposite directions in the two limbs of Henle's circle, and so is the movement of blood cells in vasa recta. You can see it clearly. See, here the movement is into the downward direction of in the descending loop of Henle, and then here it is in the upward direction in the ascending loop of Henle. In the same way for vasa recta also, see here you can see it is in the downward direction, whereas on the other side of vasa recta it is in the upward direction. Here let's talk about the concentrating urine. Sodium chloride NaCl can be transported from the ascending limb. Previously also we talked about it. It's a bit thicker limb. Sodium chloride can be transported from the ascending loop of Henle to the descending limb of vasa recta. Here it's shown the ions which are being transferred. The ascending limb of vasa recta. Now we are talking about this part. The ascending limb of vasa recta in turn carries sodium chloride to the interstitium. What is interstitium? That is the tissue between Henle's loop and vasa recta. A concentration gradient of 300 milliosmolar is thus created in the cortex to 1200 milliosmolar in the medulla. Now about urea. Urea contributes to this process by being transported through the descending limb of Henle's loop into the interstitium. Higher and higher amounts of solutes are found in the interstitium as urine flows down from the collecting tubule. Then osmosis causes it to lose water. See, you can see here all H2O. So this is how the urine is concentrated. Congratulations friends, we have successfully completed and we have learned how is urine concentrated, which is known as countercurrent mechanism that happens in the juxtaglomerular apparatus of each nephron. Hope you have learned it thoroughly. If you feel it useful, please do share it with your friends also. Wish you good luck. See you again with another very important topic. Bye for now.